We're going to begin our discussion of eyes and vision by looking at what's known as the lacrimal apparatus. Basically, these are structures involved in the production and drainage of tears. So, tears help lubricate the eyeball. They also help to flush anything that might have landed on the eyeball. And these tears are produced by what's known as the lacrimal glands. Lacrimal glands, you'll notice, are situated above the eyeball and slightly lateral. These glands respond to stimulation of the facial nerve. So, cranial nerve 7, the facial nerve, uses parasympathetic neurons to stimulate the lacrimal glands to produce tears. These tears are then drained through the lacrimal ducts onto the eye. And once on the eye, they're either going to kind of slowly evaporate or they're going to flow into what's called the lacrimal canaliculi. So, canaliculi basically means small canals. So, there are these small little canals that drain the tears. They will then drain into what's called the lacrimal sac, down the nasolacrimal duct, and then finally into the nasal cavity. All right, so tears, depending on how much tears you produce, right? You might not need to drain that much because as you produce the tears, it kind of gets evaporated off, and so you could just maybe need to drain a little bit. If you're producing a lot of tears, maybe there's an emotional stimulus that increases tear production. You'll have to drain more tears into that nasal cavity. And if these little canaliculi can't handle all of the tears, they could spill out and run down your cheeks. So that is the lacrimal apparatus and tear production. The next topic we're going to talk about is movement of the eyeballs. And you'll notice that a lot of this information in this chapter we've kind of touched on here and there because we learned about cranial nerves. So I'm going to kind of keep these muscles under their proper cranial nerve. So there's 6 extrinsic eye muscles. They all will insert and attach to the white part of the eyeball. The white part of your eyeball is known as the sclera. And these muscles will attach to that sclera, pull on that eyeball, and move that eyeball in a certain direction. So if you recall, we have the oculomotor nerve, which controls 4 of the 6, the superior rectus, which elevates, the inferior rectus, which depresses the eyeball, the medial rectus, which adducts the eye, and then the inferior oblique, which elevates and rotates the eyeball outward. The abducens nerve controls one muscle, the lateral rectus, which causes abduction of the eyeball. And then the trochlear nerve controls the superior oblique muscle, which depresses and rotates the eyeball inward. And I'll point out the trochlea, which is this little pulley that this muscle uses. So notice that this superior oblique comes from way back here at the optic nerve, goes through that little trochlea, and then angles back onto the eyeball. So there's that oblique angle. The inferior oblique is on the bottom. Again, it runs on an angle. So those are your 6 extrinsic eye muscles. We should be able to locate all of them, either on this picture or this lateral view. So you'll notice the superior rectus we can see on top of the eye. This is a superior view, so it'll be right on the top. On this picture, we see it right on the top of that eyeball. The inferior rectus we can see down here, all right, because it's below the eyeball. Since this is a lateral view, we see the lateral rectus. Don't see the medial rectus because it's on the other side. Oops. On this picture, we see both the lateral rectus and the medial rectus on each side. All right, the obliques, again, we see the superior oblique here. We also see it up here using that trochlea, and then the inferior oblique is on the bottom. Again, oblique is on an angle, whereas rectus runs parallel to the midline. So those are your 6 extrinsic eye muscles. Now we're going to use this picture to learn the structures of the eyeball. And typically what we do when we're learning the eyeball is we learn it by layers. You'll notice here in this picture we see these different layers of the wall. Then we look inside the eyeball and identify some regions, either behind the lens, back here, or in front of the lens, and a few other accessory structures. So one of the anatomical words we use for a layer is a tunic. Blood vessels have layers, so we refer to those as the tunics, and then the eyeball has tunics. The outermost tunic or layer is called the fibrous tunic. Fibrous kind of suggests a little bit. It's a very tough layer. It has a lot of fibrous connective tissue. And that fibrous tunic should be the outermost layer, so it would be this layer here, and then it continues in the front and then goes all the way around. So it's the outermost layer divided into 2 parts. In the front, or the anterior side, you have the cornea, and then everywhere else where it kind of turns from clear or transparent into white, that white part is the sclera. All right. So what's the function of these parts? The cornea does 2 things. It admits light, which basically means lets it into the eyeball, and it admits the light because it is transparent. All right. And then it does some refraction. Refraction is a very important process to understand. It's bending of light rays. So it's not reflection. All right. If you reflected the light, it would reflect away. But when the light goes through the cornea, it bends. All right. And that bending of light is very important in order to be able to see nice, crisp, clear images. The sclera is more of a protective layer. It helps maintain the shape of the eyeball. It also protects inner structures. All right. So fibrous outer layer divided into either cornea in the front, and it's going to be clear, transparent, and sclera is the white of the eye. Then we move in and we find another layer. So now we pass through the sclera and we see a layer here. All right, and this layer is often referred to as the vascular tunic because it has a lot of the blood vessels that enter and have to distribute blood throughout the eyeball. Now, the vascular tunic is going to be divided into 3 parts, and they're all pretty continuous with one another. So they're all part of this middle or vascular tunic. So we're going to first look at the iris. So the iris is basically that part of the eye that you see in people that, gives people different color eyes. So there's some pigmentation with the iris. You could have blue eyes or brown eyes or green eyes or hazel eyes. And that's the iris. And I'll remind you that the iris is a muscle that controls the hole that's in the center of it. And that hole is known as the pupil. All right. This iris is made up of smooth muscle. All right. So it's involuntary smooth muscle, and it's controlled partially by the oculomotor nerve, which provides the parasympathetic neurons, but it also has sympathetic neurons going towards it or into it. So that's the iris. You'll notice it is continuous. So if we look at it here, it continues and forms this structure, and then it continues and forms all this. So that's the 3 parts of the vascular. Iris, then we go ciliary body, which is this structure here, and then you can see it up here as well. The ciliary body has 2 major functions. One is it secretes a liquid called aqueous humor. Aqueous humor fills this portion of the eyeball that's in front of the lens. This is known as the lens. Everything in front of that lens, there's a liquid. It's very watery, so it's aqueous. And the word humor means body fluid. We don't use that word that often, humor, but it doesn't mean humorous. It's not funny. It has nothing to do with the bone, the humerus. A body humor is a fluid. You could call blood a humor. You could call lymph a humor. Your eyeball has two humors. It has one up here called aqueous humor, and then it has this material shown kind of as kind of purple is the vitreous humor. All right, so, and we'll talk more about what this humor is for. It basically supplies oxygen and nutrients to the cornea as well as the lens. So the cornea and lens have to be clear, right? They have to be transparent. You can't have blood vessels in there. So in order to replace the blood vessels, you need some other fluid that gives these structures oxygen and nutrients. And that's aqueous humor. So that's one function. The other one, you'll notice it has little fibers called suspensory ligaments that attach to the lens. So the other function of the ciliary body is to adjust or alter the shape of the lens. And that's so you can bend the light either more or less. So the cornea you can't really adjust. It's going to bend light the same no matter what. But the lens you can adjust, okay, and that's called accommodation. Accommodation of the lens is adjusting the curvature so you can bend the light more for near vision or bend the light less if you're using your far or distant vision. All right, and we'll talk more about that as we go forward. So that's the ciliary body. Then if you continue, it's going to kind of transition into what's known as the choroid. And the choroid basically is this layer in between the sclera and the retina. All right, so sclera again is the white. This yellow is the retina. So choroid is in between those two. It provides blood flow. It also will absorb any scattered light that might be in the eyeball. So it has some pigmentation that absorbs light. So vascular tunic is the layer that, there's quite a bit there. The iris, ciliary body, choroid. These structures might do more than just one thing, but these are part of the puzzle, right? We got to learn all the pieces first before we can learn how an eyeball works. All right, so then you got the inner layer. The inner layer we often refer to as the nervous tunic or the neural layer because it's got neurons. All right, and that's your retina. Okay, so the nervous tunic is the retina, and it's all the yellow. So it kind of starts up here and continues all the way to the back and goes all the way here. So it's kind of like a inner kind of cup shape. All right, it's almost like it's catching the light that comes in. So light will travel in and hit the retina. All right, so it's that inner surface that's mostly in the back of the eyeball. It doesn't go all the way into the front, okay? So notice it starts here and basically the same area here and goes all the way in the back, all right? The retina has different layers. One layer is going to have the photoreceptors. So those are going to be the sensory neurons that detect light, right? It's photoreceptor. There's two categories those belong to. You're either rods, which are more useful when there's low levels of light. And then there are cones, which are more for color. All right, so we've got our low levels of light, kind of our night vision, and then we have our color vision with our cones. And when light hits these rods and cones, it converts it to action potentials, and then those action potentials travel out of the eyeball in the optic nerve, cranial nerve 1. All right, so those are the layers. That optic nerve, by the way, is not photoreceptors. So the photoreceptors are only in the retina. But then those photoreceptors have to then stimulate other neurons that transmit. All right, the transmitting neurons that are found in the optic nerve are called ganglion cells. All right, ganglion cells are just as important as the photoreceptors. We know this is true because with glaucoma, the ganglion cells die. and that causes blindness. All right, so those are the 3 layers. Now, what else is there? Well, we've got to learn about the vitreous chamber. So the vitreous chamber is basically everything behind the lens and in front of the retina. So this whole container or chamber here is known as the vitreous chamber, and it's filled with a material called vitreous humor. Sometimes it's called vitreous body. So this is that other humor inside the eyeball. And it's not watery like the aqueous humor. It's more like a gel. But it's very transparent, all right, because light has to travel through it. So light has to travel through the cornea, through this anterior cavity, which contains the aqueous humor, through that pupil, which is a hole in the iris, through the lens, and then into the vitreous chamber, and then eventually hit the retina. So anything that light is traveling through needs to be transparent. So the cornea is transparent, all this aqueous humor is transparent, the lens is transparent, and the vitreous. So what's the function of the lens? The lens does refraction as well. So remember, cornea refracts light, but you can't change it. It's always going to refract basically the same way. The lens, you can manipulate it. You can change it with your ciliary body and bend the light more if the object is close, or bend the light less if the object is far. Anterior cavity contains the aqueous humor. Again, that's all in here. All right, that aqueous humor was produced by the ciliary body, and that aqueous humor supplies oxygen and carbon dioxide to the lens and the cornea, because the lens and the cornea do not have blood vessels. They are avascular. All right, if this material builds up, aqueous humor, it causes a high pressure, and the pressure kind of goes high in the whole eyeball. And a high intraocular pressure is a risk factor for glaucoma. So if you've ever had that test where they shoot a puff of air at your eyeball, that's testing the pressure inside of the eyeball. And high pressure has been linked to killing the ganglion cells and leading to glaucoma. And then again, the vitreous chamber contains vitreous humor. The function of that vitreous humor is just to put a little pressure up against that retina and prevent it from detaching. All right, so this gel puts just enough pressure on that retina so it doesn't peel off. It also helps maintain the shape. So it maintains shape of the eyeball and keeps that retina from detaching. All right. So there's some of these other structures. So other than that, we're good. We're as far as the structures. Now, we also want to maybe take a couple other views here. So this is another view of the eyeball where now we're looking at all the muscles that are, helping to move the eye. There's the inferior rectus. There's an oblique, inferior oblique. Above the eye, we can see the superior rectus. We see the levator palpebrae superioris. Now, notice that this muscle is going right into that upper eyelid, so it can elevate that upper eyelid. Whereas this muscle here, superior rectus, is attaching to the eyeball itself. There's another muscle here I'm not going to hold you to, this superior tarsal muscle. That can also open up those eyes significantly. It's kind of more in response to like a fight or flight situation where, if you were to get in a car accident, your eyes would open up, a lot more and involuntarily with that tarsal muscle. Orbicularis oculi, remember that muscle? It surrounds the eye, so that one's going to be kind of right here and down here. We see the cornea, all right, transitioning into the sclera. We see the iris right here, and we see that hole in the center, the pupil, and then the lens, and then the vitreous chamber back here. All right, so just another picture to work on the anatomy of the eyeball. How does the iris work? Well, two things that we can do with the iris, because it has two slightly different muscles. It has a circular muscle that if you stimulate it, it will constrict. All right, so this is called a circular sphincter muscle. And that's the one that's controlled by the oculomotor nerve. And that oculomotor nerve uses parasympathetic neurons to constrict that pupil. That's typically going to be in response to bright light. So we can reduce the amount of light entering the eyeball by constricting that pupil. We use sympathetic neurons to dilate it. So the sympathetic neurons control what's known as the radial muscle or the dilator muscle. So that muscle is out here. So notice here it's relaxed. All right. And then if you contract that radial muscle that kind of radiates out from the circular muscle, it'll open up that pupil. All right. So the pupil constricts, parasympathetic, bright light, oculomotor. It dilates, sympathetic, in low levels of light. So now we can let more light into the eyeball. Remember, if you have third nerve palsy or an oculomotor problem, it can't constrict the pupil. So the pupil will be kind of stuck dilated. And if you shine a light in it, it would have a hard time constricting. So that's the iris and the pupil. Retina. Let's take a little closer look at the retina. So what we're looking at here are layers to the retina itself. So here would be kind of taking a chunk out of the eyeball. We're just looking at layers of the retina, and then we finally would hit the choroid back here, and behind it would be sclera. All right, so remember you got sclera, choroid, and then retina. But notice the retina has different layers. We're not going to go through all of them, but notice that you've got a number of layers that are kind of in front of your rods and cones. So the light, when it travels into the eye, it actually has to travel through a couple layers of neurons in order to get detected by your rods and cones. So this is the photoreceptor layer of the retina. Oops. All right, so light travels this way through, gets detected by your rods and cones. When they get stimulated, they transmit this way and then that way. And then eventually these fibers of the ganglion cell form that optic nerve. All right. These rods and cones, again, the rods are going to be for low levels of light. So we often refer to that as kind of our night vision, but also shapes and shades of black and grays. Whereas your cones come in three flavors. You've got your red cones, your green cones, and your blue cones. So they're much better in the bright light. So you can see color much better with bright light. And they provide our best visual acuity. Visual acuity is the sharpness of vision. All right, so cones are really good for your nice sharp vision. That's why at night you don't have very sharp vision, because you're working primarily with your rods. All right, so that's the retina. What's the take-home message here? Know what your rods and cones do. Know that light actually has to travel through a couple layers of neurons to get detected by the rods and cones. Then there's a layer behind the rods and cones called the pigmented epithelium. These are going to be helpful in maintaining the health of these rods and cones. This is what the retina would look like if you shined a light in someone's eye and looked in there. We would be able to see the central part of the retina known as the macula lutea. And then within that macula lutea is the best part of your retina. It's called the central fovea. The central fovea, it contains your highest concentration of cones. So if you want to see something really, really well, you look at that object, right? The reason why you have to look at that object is the light will then hit your central fovea that's within the macula. All right. If the image hits your outside your macula and your central fovea, that's your peripheral vision. So your peripheral vision is not as sharp as your central vision. And that's because you've got less cones in your peripheral vision. All right. So again, cones give you the best visual acuity. You have the most cones within this central part of your macula called the central fovea. Off to the side is your worst spot. It's called your blind spot or your optic disc. That's where the optic nerve is. So any light that hits this optic disc, you can't detect it because you have no photoreceptors. All right, so there are no photoreceptors at the optic disc, and therefore it's often referred to as your blind spot. Now, the beauty is you got two eyes. So whatever light hits the blind spot in one eye does not hit the blind spot in the other eye, so you don't see a spot, right? If you close one eye and look around, you still don't see a blind spot because you have a brain. Your brain tricks you. All right, your brain basically allows you to not perceive a blind spot. All right, by kind of filling that in with surrounding information, and you have no idea that there's actually a blind spot there. All right, so kind of interesting. Okay, this just shows why you have a blind spot. So this is showing exactly where that optic nerve comes out the back of the eye. So if light is traveling in the eye and hits right here, there's no photoreceptors. Notice here are all photoreceptors here and here. So because you have all these ganglion cells that have to transmit out the back of the eye and form that optic nerve, there's no room there for photoreceptors. All right, so that's the optic disc. Also notice that you've got blood vessels that come through that optic nerve at the optic disc. All right, vision. So let's talk about kind of your basic principles. One major principle is refraction. So you'll notice when you're looking at an object, all right, so this tree is off in the distance. The light that comes off of that eyeball has to be bent. All right, it's bent by the cornea. And then it may or may not have to be bent much using the lens. All right, the object is actually flipped upside down when it hits your central fovea, but you don't perceive an upside down tree because you have a brain. All right, your brain knows that, wait, we have to perceive it the other way. Now, what if that object was close? Like a letter. So now you're looking at text that's very close up. The light rays that come off of that are diverging, all right? So they're kind of going away from that object. So now those light rays have to be bent more. So notice what we have to do to the lens. We have to make it a little more rounded to refract even more, all right, in order to see that object. So your eyes are basically set for distant vision. You don't have to do a whole lot of adjusting if you're looking at something in the distance. But if something ends up coming closer, we have to adjust the lens. We have to make it a little more rounded, and that's done using your ciliary body. All right. So refraction, super important. Notice that your light is bent actually in 3 places. So when light goes from air to cornea, it bends once there. When it goes from aqueous humor into the lens, it bends once there. And then when it comes out of the lens into the vitreous, it bends a 3rd time. So notice it's best shown right here. So this light rays come here, they bend once in the cornea. They bend again when they enter the lens, and then they bend again when they exit the lens. So there's actually 3 interfaces that do the refraction of light. But it's basically 2 structures doing it. It's the cornea, and then it's the lens. But it bends when it goes into the lens, and then it goes out of the lens. So that's refraction. We should understand that the cornea does it, and your lens does it. But with the lens, we can adjust it based on whether we were looking at a distant object or a close object. And again, that's called accommodation. So that's one of the adjustments that the eyeball can make. It can change the shape of the lens. The second adjustment is constriction. So we can restrict the amount of light over the area of the greatest bulge of the lens. So we can kind of adjust using our iris and pupil. We can restrict the light where it's going to go through the lens, the best location. And then we also can converge. So since we have two eyeballs, we can converge on an object. All right. So for example, if there's an object, let's say there's a bug on the tip of your nose. Well, you know that your eyes are converging onto that object. All right, disorders. That brings us to some of these disorders. So we're going to first talk about disorders of refraction, because there's a lot of eye disorders out there, and they're not all because of improper bending of light. But there are a few that are due to refraction. All right, the three we're going to talk about are myopia, hyperopia, and astigmatism. So myopia is often called nearsightedness. All right, so when someone is nearsighted, they can see close objects fine, but they have trouble seeing distant objects. And the reason is that the focal point, so after the eyeball does all the bending, the focal point is premature. It's in front of where it should be. All right, so in essence, you're bending the light too much. All right, so that could either be because the shape of the eyeball is off. All right, so the eyeball might be too elongated, or the lens or cornea might be too thick. All right, and it bends the light too much. The good news is we can fix it by using a concave lens. So notice a concave lens bends the light out a little bit. All right, so now when those light rays come in, we've bent them out a little bit, but now when the eye does its thing, the focal point is right on that central fovea. All right, so that's the corrected version of myopia. This is if we did not correct it. Hyperopia is the opposite. So now the eyeball might be a little short, or the lens or cornea might be a little too thin, and the light rays are not bent enough. All right, and that causes hyperopia or farsightedness. So they can see distant objects fine, they have trouble with close objects. How do we fix it? We use convex lenses. So the convex lens will actually refract the light more prior to it entering the eyeball. And then finally, astigmatism is unequal curvature. So some parts of that cornea or lens are curved differently. So now in someone's field of view, some areas are blurry and some areas are not. So the key with astigmatism is kind of this irregularity or unequal curvatures. What are some other eye problems or forms of blindness? There's a number of them. Color blindness, that's a genetic. It's called X-linked genetic, and X-linked diseases are more common in males because males only have one X chromosome. So if you have a genetic defect in a gene on the X chromosome, there isn't another X chromosome that can help. All right, you don't have another copy of that gene. Whereas in females, they've got two copies. So it's very rare to have the defect in both copies of the gene. Cataracts is cloudiness of the lens. That can happen with age, infection, trauma, and that's a very, very common cause of blindness. Macular degeneration, as the name suggests, is where the central part of the retina degenerates and you lose sharpness of the central vision. So your peripheral vision is okay, but your central vision is affected. And again, the problem with that is your central vision is your sharpest vision. So these people have a very hard time maybe reading because that requires very sharp vision. But their peripheral vision is fine. There are some other forms of it. There's some early onset macular degeneration. In fact, my younger son has a form of macular degeneration, and it's a genetic defect where he's losing his central vision early in life. Glaucoma is a major cause of blindness. That again is due to high pressure, often due to the buildup of aqueous humor, and then it's the loss of ganglion cells. And then finally, there's diabetic retinopathy. There's a lot of problems there. Optic nerve can degenerate. You can get cataracts formation, retinal detachment, hemorrhaging, and blood vessel degeneration. So those are some forms of blindness. The next video, we will get through the ear. Again, with the ear, we have to separate out structures that are involved in hearing from the structures that are involved in equilibrium.